Hello and welcome to Bacteria. We're going to dive a little bit deeper into how bacteria cause disease. And after you've watched this video, you will understand the extent, origin, and functions of the human microbiome. You'll appreciate the ways that bacteria enter and spread in their hosts. You'll be familiar with the different types of bacterial toxins and their activities. And you will know the mechanisms of action of members of the different classes of antimicrobials and how resistance emerges. Before we talk about bacteria that cause disease, it's very important to first discuss the human microbiome, all of the microbes that live in and on us. We're home to trillions of microbes, trillions, and that's what we call the microbiome. And it's a composition of all the microbes that are everywhere in us and on top of us. A human body is only 25% human cells. Now look, you can see me. You think you're looking at just human cells. 75% of what you see here are bacteria. Very, very little human cells. Bacteria, fungi, and archaea. It's amazing. Wherever the human body is exposed to the outside world, there's a microbial community. The mouth, the lungs, the GI tract, the urogenital tract, the skin, and probably other places we haven't even found it yet. This microbiome is just beginning to be unraveled in terms of what it does for us. It certainly helps us to extract energy and nutrients from the food we eat, but it also appears to inhibit the growth of pathogens, not only in us, but on our skin. The microbes on our skin produce antimicrobial compounds that protect us. It's remarkable. There's probably much more that the microbiome does. For example, recently it's been shown in animals that the microbiome helps their immune system to develop. It's really, really remarkable. In the coming years, we'll be able to sort all of this out. But it's quite clear from the very early days of our existence, when we're first born, our microbiome forms, and it has a huge impact on our health. Microbes contribute an extra 2 million genes to the 20,000 or so genes that our human genome encodes. 99% of our genes are bacterial. Isn't it remarkable? Our microbiome weighs 2.5 pounds. That is weight you'll never be able to get rid of. You can try and lose some of your body fat, but that 2.5 pounds of microbiome needs to stay with you. Otherwise, you're going to be very unhealthy. And don't forget the viruses. All those bacteria and fungi and archaea in us, they also are accompanied by their viruses. In fact, viruses outnumber bacteria by about 5 to 1 in us. And it's quite clear that they have major roles in regulating the microbiome. The volume of the microbiome is about 3 pints. The next time you drink 3 pints of beers, I want you to remember you've just drunk the volume of your microbiome. It's my way of getting you to remember what I'm telling you here. Now, where do you get this microbiome? There are many ways. While you're developing in utero, you are bathed in amniotic fluid provided by your mother. And of course, it's going into your mouth and on your skin, and it's got bacteria and viruses in it. So that's your first encounter. You're acquiring your microbiome in utero. Then as you are born, you acquire more of your microbiome during birth. If you so happen to have been born by passage through the birth canal, you will start to acquire the bacteria that are present along that canal, not just in your mouth, but on your skin. People who are born by cesarean section, they don't go through the birth canal. Of course, they come out through an opening in the skin produced by surgical manipulation. They have a very different skin microbiome from children who are born vaginally. So it's probably better to be born through the normal routes. You acquire probably a more beneficial microbiome. But of course, in some cases, it's not possible for health reasons. You also acquire a good amount of your microbiome from your mother. Your mother is one of the first to hug and kiss you, and she continues to do so, hopefully, for the rest of your life. And your father also contributes, and any caregivers who may come into your home, they are all contributing to your microbiome. Breast milk is a very important contributor to your gut microbiome. Again, breastfeeding is not done by everyone, but it's been shown in many studies to be better indicators of subsequent health. You also acquire microbiome from the soil that you may touch. You should let your kids play in the soil. It's probably good for them to help acquire their microbiome from the water that you wash yourself in and drink, from the foods that you eat, and any people or pets or plants that you encounter early on in your life. The early years are formative years, and then you acquire a relatively stable microbiome that is very similar to that of your family. And only when your health changes, or if you move or change your diet, do you change your microbiome. And the consequences of the microbiome for human health are going to be learned in the next 10 years, and we're really going to find out fabulous ways that we can improve our health by manipulating it. Okay, on to infection basics. Let's talk about some principles by which bacteria can enter the host. We can divide bacterial infections into two broad groups depending on how they're acquired. Some are acquired exogenously from some external source, like bacteria in the environment, or bacteria in the food, the water, the air, the objects we touch, insect bites, or any animals, dog bites, for example. And so those are exogenously acquired from elsewhere, and that contrasts with endogenously acquired infections, where our microbiome suddenly turns on us. And this could be because we've altered the microbiome by treatments with antibiotics, and clostridial infections of the intestine are a great example of that, where we alter the composition of this family of microbes and then suddenly Clostridia overgrow and cause us problems. Sometimes injuries introduce skin bacteria into us, and this is commonly occurring with Staphylococci, normal inhabitants of the skin. When an injury introduces them below the skin, this can cause a problem. So we get bacteria that cause infections and illness from external sources and from within ourselves. Let's talk a bit about how bacteria gain entry to the host, the exogenous sources of infection. Here's a human body which provides a very large spectrum of places for bacteria to enter. A very common place is a mucous membrane. We have mucous membranes all throughout our body, our eyes, our mouth and nose, the entire alimentary tract, which is essentially a very long tube starting from our mouth, going through our intestines and out the anus. That is all lined with mucous membranes because it has to absorb food and excrete waste. And because it's a mucous membrane, it is vulnerable. It is not sealed against entry. Things like breathing, eating, having sex, all can introduce pathogens into us. Cholera, whooping cough, and gonorrhea are examples of bacterial infections that are acquired across mucosal membranes. Now, we happen to be covered by a wonderful protective organ called the skin. Skin is the biggest organ in your body. It weighs the most, and it has the most square area. And it is a great barrier. The outer layer of your skin is dead, so viruses cannot multiply in them. They have to get inside by penetration. But there are ways that the skin can be breached, allowing bacteria to invade the underlying cells and tissues. How can this happen? Insect bites, of course. They routinely deliver things below the skin. Any kind of scratch or injury, which we're all prone to having, will also breach that wonderful protective barrier. Once you're in a mucosal membrane, there are ways to get across that, which are even easier than in the skin. You typically don't need an injury. Here's a cross-section of the intestinal mucosal barrier. And on the left side is the lumen of the gut, the space on the interior. And the first line of defense is an epithelial cell barrier. And below that are the submucosal tissues. And that also contains capillaries and lymph capillaries. So if a pathogen can get across the epithelial cell sheet, it has access to the circulatory system, which can bring it anywhere. And of course, below that are the muscle cells that help to contract the intestine during digestion. If we take a closer look at this intestinal lining, we can see that there are different kinds of cells that make up that epithelial sheet. There are enterocytes, which are involved in taking up nutrients, and then interspersed among them are the so-called M cells, in which immune cells come from below, lymphocytes and macrophages, and sample the contents of the gut to make sure there isn't anything nasty there. Both the enterocyte and the M cell can be points where pathogens can cross into the underlying tissues. Once a bacteria has adhered to the epithelium, which many do, as we'll talk about later, they can move laterally, reproduce, and move across to contiguous tissues, or as I said, they can penetrate the epithelial sheet and disseminate to distant sites. Our immune defenses play a big role in limiting such incursions, but they don't always win, and bacteria do enter the rest of our body. Once a bacteria or many bacteria are in us, they typically multiply in order to cause disease. And this graph shows a time course of some typical infections. The x-axis shows you time, and the y-axis shows you various parameters, including the production of bacteria in red and immune responses. So there is a period initially after the bacteria enter us called the incubation period. This is an extremely important concept. This is a period which is variable among the different infections, during which the bacteria are multiplying, but we don't yet have overt disease. During that period, immune responses may be mounting, but again, and you may have fever as a consequence of that, chills, aches, and so forth, but you won't have the actual symptoms of the disease. That is the incubation period. Then, as the bacteria multiply, they may either elaborate toxins or they may invade and cause tissue damage. You have a period of disease, and if you follow the red line and go above the blue segment of this graph that says no disease, above the blue is where we have bacterial disease, and this can be a consequence of both the immune response and direct effects of bacterial toxins, which we'll talk about in a moment, on us. There is a very important part of this graph which shows no disease. Some bacterial infections where the bacteria are replicating will not result in disease. These are called asymptomatic infections, and many people have these. It's a consequence of them being able to control the replication of the bacteria, and others may have simply more resistance to disease formation. And the outcome, of course, of a bacterial infection can be recovery and cure and subsequent immunity. It can be, unfortunately, in many cases, death, or in some cases, immunity is not lifelong and we obtain recurrent illnesses with the same pathogens. Let's talk a bit about bacterial toxins. These are molecules produced by various bacteria that alter the normal metabolism of host cells, and they are often responsible for the major symptoms of bacterial infection. And there are many different kinds of toxins that are produced, which we'll talk about briefly. Interestingly, this is in direct contrast to viral infections, in which very few viral toxins have been identified. They cause disease in very different ways from bacteria. Now, we recognize different classes of bacterial toxins. Some are called exotoxins because they are secreted by the bacterium into the extracellular environment. These exotoxins shown on this picture have a typical A-B structure. They have subunits, separate subunits, consisting of an A component and a B component. Typically, the way they work is they're secreted by the bacterium and they bind a receptor, shown here in R, on the surface of the eukaryotic cell, on the surface of our cells. They are then taken up into the cell by the endocytic pathway, and typically the A component, which is the active component, is released from the receptor binding component, makes its way into the cytoplasm, where it then has its effect on cells. There's another class of toxins called the type 3 cytotoxins. They're shown on the right of the diagram, where we see an outline of a rod-shaped bacterium and what looks like a syringe injecting molecules into the host cell. Those are type 3 cytotoxins. They're directly injected into the host cell by a structure on the bacterium called a secretory injection system. And these have evolved just to inject toxins into the cell. These secretory systems act by introducing molecules into cells to alter their behavior. Other toxins are produced by bacteria that act at the surface of host cells. Some of them bind to pattern recognition receptors and induce the production of cytokines, which have lethal effects. There are also pore-forming toxins, which make holes in cell membranes and make them die. And finally, there are toxins called superantigens that bind to T-cell receptors and major histocompatibility receptors and induce the synthesis of many, many toxic cytokines. And finally, there are proteins called exoenzymes produced by bacteria that modulate targets in the extracellular matrix. Let's look at toxins in a bit more detail. I mentioned that they typically have an A-B structure. The diphtheria toxin consists of one molecule of the A and one molecule of the B. Again, the B is the receptor binding component. The A is the effector portion that actually has an effect on the host cell. The diphtheria toxin, the A portion, blocks cell protein synthesis. The A portion ADP ribosylates an elongation factor for translation EEF2. It stops host cell protein synthesis and kills the cell. Cholera toxin is composed of a single A subunit and 5 B subunits. This toxin elevates intracellular cyclic adenosine monophosphate in the epithelium of the small intestine, and that causes movement of fluid into the lumen and the classic diarrhea associated with cholera. And finally, the anthrax toxin, the highly lethal anthrax toxin, is composed of 2A and a B subunit, and again, the B subunit binds the cell receptor. Two toxins that are well known, botulinum toxin produced by C. botulinum and tetanus toxin produced by C. tetani clostridium, are neurotoxins. These toxins, elaborated by the bacteria at different sites, make their way through the circulatory system and the lymph system to the brain, where they cause their effects. Tetanus toxin, for example, causes muscles to contract uncontrollably, and they cause what we call spastic paralysis. On the other hand, botulinum toxin blocks muscle contractions, so the muscles get flaccid. This is called flaccid paralysis. So two very different effects on the central nervous system. The type 3 cytotoxins we mentioned briefly before, they're shown on the right-hand part of this screen. They are injected by the bacterium into the host cell by a type 3 secretion apparatus. Bacteria have a number of different kinds of secretion apparati, which are used to inject effector molecules into the host cell to get them to do what they want. And these have their own ways of altering the biochemistry of the cell to cause pathology. Type 3 cytotoxins are found in a wide range of bacteria. For example, Salmonella, Shigella, Pseudomonas, cholera, and the plague bacilli all produce type 3 toxins of various sorts, and we'll mention a few of these and how they work in a few moments. Another toxin, and a very important one, produced by gram-negative bacteria is endotoxin. Now, if you don't remember what a gram-negative bacteria is, go back to the introductory bacteria lecture and take a look. Endotoxin is sort of like the calling card of gram-negative bacteria. It announces to the host, I'm here, and you better watch out. The endotoxin is nothing more than lipopolysaccharide, that outer layer on the outer membrane of a gram-negative bacteria. So that outer layer is a lipid bilayer, but the outer layer is unusual. It's not the standard kind of lipid. It's made of lipopolysaccharide, and that is what is endotoxin. Now, despite the name endotoxin, this toxin is not internalized into the host cell. It remains extracellularly. It just happens to be the name that has stuck over the years. Let's look in closer at that outer membrane, that lipopolysaccharide. Here it is in some detail. Remember, at the very bottom, there are lipid chains that form the outer leaflet of that membrane, and that component is called lipid A. That's the active component of endotoxin. That is what has the biological effect. And then there are other portions, including the O portion that we talked about, which extends above, completing lipopolysaccharide. Lipopolysaccharide is recognized by pattern recognition receptors located on the surface of the eukaryotic cell. We all have such receptors in order for us to recognize what is foreign. And when foreign molecules are recognized, the result is a production of cytokines, which mobilize the immune response, but may also have detrimental properties. So on the left of this slide are 3 pattern recognition receptors, one of which recognizes LPS, that portion of the outer membrane of bacteria that we've just been talking about. Another one, FLA, recognizes flagellin, the protein that makes up the flagella of bacteria that helps them to move. So here's a close-up look of the pattern recognition receptor, and it again recognizes endotoxin as being present, initiates cytokine synthesis, and depending on how much endotoxin is present, low or high, it has different effects on the cell. So you can imagine that early in infection, it has one kind of effect where the endotoxin is low, and then if the infection proceeds unchecked, you'll have higher concentration of endotoxin, you'll have a very different effect. These receptors for endotoxin and other bacterial products are called innate receptors. They also sense viral infections as well. So at low concentrations of endotoxin, there are a variety of effects, many of which reflect the attempts at the immune system of eliminating the bacterial infection. So let's take a look at some of these. You can see the 4 different targets here and the ultimate activities and effects of endotoxin. At low concentrations, endotoxin targets Kupffer cells, macrophages of various sorts. It causes release of cytokines. When it binds to its receptor, cytokines are produced, and some of those cytokines cause fever. So endotoxin is well known to be pyrogenic, fever-inducing. The mechanism is by being recognized by that innate receptor and producing a cytokine that induces fever. Cytokines, endotoxin also activates macrophages. It makes them more phagocytic. It makes them secrete hydrolases and have just more enhanced killing. The idea being, of course, there's endotoxin here, we sense it, there must be a gram-negative, we're going to kill it. So we're going to get activated. At low concentrations, endotoxin also activates neutrophils. This has an effect of dilating blood vessels and allowing immune cells to come in and clear any infections that may be present. Endotoxin also activates B lymphocytes. B lymphocytes, of course, produce antibodies, and these may be useful for clearing the infection. Finally, at low concentrations, endotoxin also activates the complement system, whose goal is manyfold, essentially trying to get rid of the bacterial infection. Complement can help take up bacteria into macrophages by opsonization. It can increase capillary permeability, and it can also poke holes into bacteria. All of this is part of the inflammatory response to infection. Now, in contrast, at very high levels of endotoxin, it often can result in shock, fluid loss, for example, caused by too many cytokines, and disseminated intravascular clotting or coagulation. There are other membrane-damaging toxins as well, lipases, for example, enzymes that digest lipids. An example is the lecithinase from Clostridial species. This enzyme can lyse cells and eliminate defenses and provides nutrients for bacteria. These are bacterial toxins lysing eukaryotic cells to avoid defenses, and the lysed cells produce nutrients for the bacteria. Hemolysins can lyse red and white blood cells. And then there are toxins that form pores in the cell membrane. They insert into the membrane, they allow water to flow in, and the cell bursts. Again, a way for bacteria to avoid some of those immune cells that are trying to get rid of the bacteria. We also have what are called heterogeneous pore-forming toxins. These are produced by a variety of bacteria. One well-known one is streptolysin O, produced by the Streptococci. This pore-forming toxin binds cholesterol and damages liposomes in cells of the host, causes the cells to lyse, part of the reason why tissues are damaged. Another set of toxins that are produced, and if you're thinking, boy, bacteria make a lot of toxins, you're right. This is their modus operandus. These are called extracellular matrix toxins. The extracellular matrix is the area between cells. Here on this picture, we're showing two cells, and the area between them and below them. This is filled with all kinds of substances that provide protection and hold the cell together. This is the extracellular matrix. Bacteria produce enzymes called hyaluronidase. Hyaluronic acid is a component of the extracellular matrix, as you can see here, and this breaks it down. It breaks down connective tissue, allowing bacteria to spread better. Streptokinase is an enzyme produced by Streptococci. It activates plasminogen, converts it to plasmin, which then attacks blood clots and gets them to dissolve. Bacteria don't like blood clots because it inhibits them from moving about and may restrict them. So this enzyme takes care of that. And finally, collagenases and elastases also digest the extracellular matrix, allowing free flow movement of the bacteria. I'd like to end up this discussion by talking about antimicrobial compounds that are used to treat bacterial infections. And think, we have a bacterial infection in us. We need to treat them with drugs that will kill the bacteria but will not harm us. In other words, the antibiotics or antimicrobial compounds have to be selective. They have to target things in the bacteria that are not present in our cells. Fortunately, this is relatively easy to do because bacteria are very different from eukaryotic cells. I'll give you an example of such selectivity. There is a class of antibiotics called the beta-lactam antibiotics, and these include the penicillins, the cephalosporins, and the carbapenems. They're called beta-lactam antibiotics because they have a chemical ring in them called the beta-lactam ring. These antibiotics target the synthesis of murine. Now, do you remember from an earlier lecture what murine is? In gram-positive bacteria, this forms a thick layer on the outside of the bacterium just above the cell membrane. And it's composed of sugar molecules joined together and cross-linked by amino acids of short length. On the right of this slide is a diagram of the synthesis of murine. Murine is exclusive to bacteria. It does not occur in eukaryotic cells. And on this slide are 4 different antibiotics: phosphomycin, cycloserine, vancomycin, and penicillin, which block different steps on the synthesis of murine. So these antibiotics work beautifully because murine is only in bacteria and not in our cells, so they have very, very little toxicity. And you can see we have developed over the years antibiotics that target different steps in the synthesis of murine, including penicillin, that last step, which is assembling the cross-linking amino acids between the sugar chains. Unfortunately, as we develop new antimicrobial compounds, resistance to them rapidly emerges. And today, we have resistance to almost every antimicrobial compound that we have developed, and the situation is becoming dire because we have fewer and fewer options with which to treat bacterial infections. Antimicrobial resistance occurs in nature, and it's ancient. Bacteria make antibiotics to compete with each other in nature, and many of those we have harnessed to use as antimicrobial treatments of infections of people. We know that these genes that confer resistance have been around for thousands and thousands of years. We can find them in very old sites on Earth, and there's plenty of evidence that they existed way before humans developed antimicrobial compounds. So we're really taking advantage of something that exists in nature. There are a number of mechanisms by which these antimicrobial genes, or I should say antimicrobial resistance genes, work. For example, they may direct the synthesis of an enzyme that breaks down the drug, a simple way of doing resistance. They may chemically modify the drug so it interferes with its function. They could inhibit the uptake of the drug into cells and tissues so it can no longer access its target. Or they could stimulate the export of the drug from the bacterial cell so it's no longer bactericidal. Or they may modify the target site of the drug. So there are many different mechanisms of antimicrobial resistance. And again, these are all encoded on genes that code for proteins that have these various activities. Let's take an example to illustrate that. And we'll use an example, the antibiotic vancomycin. Its target is cell wall modification. So vancomycin acts by blocking the assembly of the murine cell wall. Now, at the top of this slide is the normal incorporation of the precursors of murine. So the blue and the green ovals, those are sugar molecules that are going to be part of the growing peptidoglycan chain, and the smaller ovals below them, those are amino acids that will eventually cross-link the murine to make it very strong. So the way this works is that subunits are added to the growing chain. In the second part of this, in the second row of this diagram, you can see the growing polypeptide chain. Vancomycin binds to the precursors by binding to the amino acid. Vancomycin is shown here in purple with the V, or maybe that's brown, and it's binding the amino acids and blocking the incorporation into the new chain. Therefore, this inhibits murine synthesis and kills the bacteria. Resistance to vancomycin, one mechanism of resistance, is simply that the bacterium changes the D-ALA to D-ALA-D-LAC, and lactose can be incorporated into this chain. It evades vancomycin resistance, and the antibiotic no longer works. That's one example of the way resistance works. Going back to our beta-lactam antibiotics, which I mentioned before, and the arrow points to the beta-lactam ring. That's common to all members of this class. That's why we call them beta-lactams. We have so far identified over 300 beta-lactamases. These are enzymes that cut that beta-lactam ring. And these beta-lactamases encode resistance to the beta-lactam antibiotics. So you can see the extent of the problem. Beta-lactamases are everywhere. Further complicating antibiotic resistance is that the genes encoding resistance factors, for example, that encode beta-lactamases, are often able to move from bacterium to bacteria. One way to do that is via plasmids. And in fact, many of these antibiotic resistance genes are encoded on plasmids. This diagram, which we saw previously in one of our other lectures, shows how plasmids can move from one bacterial cell to another. On the upper left is a bacterial cell with a chromosome in green and a smaller plasmid in red. Let's say this plasmid encodes a beta-lactamase, which confers resistance to beta-lactams to that bacterium. Well, in the second set of bacteria, the two bacteria are now exchanging DNA through a pilus that's joining the two cells, and the plasmid is moving from one cell to another. The result is that that second cell now acquires antibiotic resistance. So, a problem here is that we often feed our animals that we eat for food lots of antibiotics so they grow quickly. The effect is that we select for antimicrobial resistance in the animals, and then when we eat these foods, we acquire antibiotic resistance genes in us, which are of no consequence initially, but then when we go to have surgery and we need antibiotic therapy, it doesn't work because we have the resistance already in us. So these antibiotic resistance genes can move around bacteria extensively. This is why they're a problem, not just by plasmid mobility, but by also movement by transduction, the exchange of pieces of DNA by viruses, or simply by naked DNA. So gene transfer among bacteria, we call this horizontal gene transfer, is widespread and is a big problem for antimicrobial resistance. And let's end up with a chart showing you some common mechanisms of resistance to antimicrobial agents. For example, the penicillins and the cephalosporins are hydrolyzed by beta-lactamases, which we mentioned. These resistance genes are in fact carried on plasmids. Methicillin resistance is a change in the penicillin binding protein, not in a beta-lactamase, but in a separate protein. This happens not to be carried on a plasmid. Tetracycline resistance encodes a pump that pushes the drug out of the bacterial cell. This is a plasmid-borne resistance factor. So if you look at all these various mechanisms of resistance, modification of the drug, synthesis of alternate substrates, and so forth, acetylation, change in binding sites, look how many are encoded on plasmids. And that simply means that they're easy to go from bacterium to bacterium. And we have a hard time treating bacterial infections when these resistance genes are so mobile. Hello, and welcome to Bacterial Infections. Today we're going to talk about Staphylococcus and Streptococcus. And when you listen to this lecture, you'll be able to know the structure and virulence factors produced by the Staphylococci and the Streptococci. You'll be familiar with the pathogenesis and the epidemiology of diseases associated with Staphylococci and Streptococci. You'll know which drugs can be used to treat staphylococcal and streptococcal infections, and you'll become acquainted with the available streptococcal vaccines. Let's dive into Staphylococci first. These are causative agents of abscesses and toxin-mediated diseases. Here we have a photograph of Staphylococcus aureus. These are gram-positive cocci, and they're often called grape-like clusters. Don't they look like grapes to you? Purplish. So that's the morphological appearance of Staphylococci. Staphylococci colonize the nose and other mucous membranes and the skin of 30 to 40% of humans. 30 to 40% of people are carrying these bacteria as part of their normal flora. Not everyone, all right, but some of them do. In your nose, mucous membranes, even on your skin. These bacteria, nevertheless, under some situations, can cause a variety of diseases. They include focal abscesses. And what we mean by that is an abscess in a very specific place, like on the skin, where it would be a boil. Many of you may have had boils before, focal infections, a very raised pus-filled lesion. We'll see some of these in a moment. But these can also occur inside of you, not just on your skin. For example, in your lungs, bones, other organs, kidneys, and heart. A general feature of the Staphylococci is that they secrete potent exotoxins. We talked about exotoxins in a previous lecture on bacteria. These are proteins that are produced and elaborated from the bacteria that have effects on the host. In the case of Staphylococci, these include the toxic shock syndrome toxin, staphylococcal scalded skin syndrome, and food poisoning. These are all a consequence of the production of toxins. The toxic shock, the scalded skin, the food poisoning are all consequences of toxin production and their effects. So among the toxins that Staphylococci produce are lipases and hydrolases. These have as an effect to degrade the lipids of your skin, and that in part contributes to the production of these boils. These Staphylococci can spread from person to person via aerosols, respiratory spread produced by coughing or sneezing or even talking. Did you know that as I'm speaking now, I'm actually making an aerosol? And so if you were standing right in front of me, you could be infected. And in particular, letters like P, where I'm sending out puffs of aerosol, that's one way that people can spread. I don't have to sneeze or talk or cough. So person to person by respiratory aerosols or by direct contact, these bacteria can be transmitted. Of course, if you already have the Staphylococci, this is not relevant. But as I said, there are many people who don't have them, and they can acquire them from others by these routes of transmission. Now, as I said before, they're on a certain percentage of humans on the skin, and they remain there harmlessly, typically, and they don't penetrate unless there's damage to the skin. You have a cut that pushes the Staph aureus deeper, and it can then cause an infection, or in a mucous membrane as well. If you have a cut in the inside of your mouth, it may introduce the bacteria into deeper tissues where it can then cause a problem. So if they're left in their normal places, they're okay, and problems arise when there are damages occurring. So things like burns, wounds of various sorts, lacerations, even an insect bite, certainly surgery, variety of skin diseases, all these conditions can let Staph aureus come in. When you have surgery, one of the first things they do is to extensively wash the skin where you're going to be cut with a preparation to sterilize the area or reduce the number of bacteria. If that's not done properly, the Staph aureus will be introduced into the wound, and you can have serious infections of the sort that we're going to talk about. So let's look at an abscess. Here's a photograph of a man. You may think that this is, in fact, a nipple, but it's not. It's an abscess caused by a staphylococcal infection. The nipple is a little bit south of this boil on his chest. So this is an example of a boil caused by a staphylococcal infection. Many people have had these. Here, the infection remains localized. It is in this boil, and it doesn't go beyond that. This boil contains pus, and that's called an abscess or a furuncle. Interesting word. Furuncle is one boil like this. A carbuncle is when you have several boils together. Makes a larger structure. Furuncle, carbuncle. So this is a typical boil. We call them colloquially, or an abscess. When the bacteria spread beyond the initial boil in subcutaneous or submucosal tissues, this is called cellulitis. So the staphylococcal infection, when it spreads beneath the skin, cellulitis. As you might predict, one of the main immune defense cells important for regulating Staphylococcus replication and spread are neutrophils. These maintain a balance of the bacteria in normal people, and if you're neutropenic, that can be a problem in controlling staphylococcal infections. Very early in infection, so let's say you have a cut, and that introduces the staphylococci, which you happen to have on your skin, into deeper tissues. There's a battle between the bacteria and your immune system involving neutrophils and other cells. Early in the infectious process, many neutrophils are killed by the bacteria, and these release lysosomal enzymes. The neutrophils are full of them. It's part of their armamentarium for killing bacteria. When the neutrophil dies, they're released into the tissue, and those enzymes then damage your tissue, and they contribute to the formation of a boil. Let's take a little bit of a deeper look at what this boil actually is, or abscess in more precise terminology. So here is a diagram of the abscess on this slide. An abscess is walled off with a capsule made of fibrin. This is a host reaction to the bacteria. The host makes a fibrin capsule to try and restrict the bacteria to this one place. Now, the consequence of this is that the bacteria are also isolated from the immune response. So it's actually a consequence both of host and bacterial action. Within this boil or abscess, we have dead neutrophils, which are killed by the bacteria, and this produces the pus. We have epithelial cells, we have bacteria, and fluid. And so this grows to a certain size. It's painful to a certain extent, and that's what an abscess looks like. Staph aureus makes a number of what we call virulence factors. These are toxins and other proteins that are made that help it to spread and overcome host defenses. And there are a number of them. In the center of that slide, there are the staphylococci, and you can see elaborating a variety of proteins like leukocidins, hemolysins, protein A, coagulases, and proteins that help make fibrin clots dissolve. Let's look at some of these in some detail. Let's take a look at some of the virulence factors produced by Staphylococci. These are gram-positive bacteria with a thick outer wall of peptidoglycan, and surrounding that is a capsule, which you may remember from our basic discussion of bacteria. The capsule is important for inhibiting phagocytosis. In this picture, we have a macrophage trying to take up the Staphylococcus, and the capsule will inhibit that reaction. It will resist it because, of course, once the bacteria is inside of the cell, it could be destroyed. The outer surface of the bacteria just below the capsule consists of peptidoglycan, which you may remember is alternating chains of carbohydrates cross-linked with short peptides. This product of the Staphylococcus activates complement. This is a host defense against infection. And complement will have various inflammatory procedures. It will cause lysis of the bacteria and production of cytokines. And so this is part of the inflammatory reaction that goes on inside the carbuncle. The bacteria also make a protein called protein A. This is quite an interesting protein. In the diagram, you can see the protein A is on the surface of the bacterium. It's shown in that little green box. What this protein does is bind the Fc portion of antibody molecules. So, antibody molecules are Y-shaped molecules. The Fc is down at the bottom, and the antigen binding sites are at the top. Normally, your antibodies would bind the Staphylococcus, but the Staphylococcus is binding the antibody the other way, so it can't bind the bacterium. And this reduces the ability of the bacterium to be taken up into macrophages by opsonization. Very clever strategy for avoiding that process. The catalase is produced by Staphylococci. This is an enzyme that cleaves hydrogen peroxide. That's hydrogen peroxide, H2O2, shown right there. Hydrogen peroxide is produced by cells like macrophages. Its product is to destroy bacteria. Bacteria, in turn, make an enzyme called catalase, and this will cleave the hydrogen peroxide to evade destruction. Very clever. The bacteria also produce an enzyme called coagulase, which converts fibrinogen to fibrin, and that helps to make a clot that surrounds the boil and isolates the bacteria from the immune response. And Staphylococci also make pore-forming toxins like hemolysins, leukocidins, and hyaluronidases that digest the extracellular matrix. All of these in an effort for the bacteria to spread and avoid host defenses. And we've talked about many of those before. Staphylococcal infections are very difficult to treat. Antibiotic resistance has become widespread among the staphylococcal strains that colonize us. There are many beta-lactamases in these, as we discussed in another lecture on general bacteria, and those beta-lactamases encode resistance to the penicillin-type antibiotics. This resistance is widespread. In addition, the bacteria make a penicillin-binding protein 2A, which further makes them resistant to penicillin-class antibiotics. And you may have heard the term MRSA or MRSA to describe these bacteria. This stands for methicillin-resistant Staph aureus. And this is very popular in the press to talk about MRSA as a superbug because they're extremely difficult to treat, almost impossible to treat with antibiotics. So MRSA describes the most resistant of these strains of Staph aureus. So these are serious infections. What kind of other diseases do these cause? They cause staphylococcal scalded skin syndrome. Try and say that 4 times quickly. This is a skin illness, as you can see here, and I think the name tells you what it looked like. This mainly affects neonates. This is caused by sloughing of the skin. The skin is coming off. It's red and angry looking, caused by toxins produced by the Staphylococci, specifically exfoliative toxins A and B. Exfoliation, the loss of your skin. You can also describe a disease called toxic shock syndrome, TSS, which became popular not very long ago. This is accompanied by fever, skin rash, hypotension, low blood pressure, and also peeling of the skin, exfoliation. Toxic shock syndrome was first described in connection with the use of highly absorbent tampons that allowed organisms to proliferate. Now, tampons went through a revolution in their production where they became extremely absorbent and also would block off the vaginal canal. And the makers thought this was a great thing because it's convenient for women, but it turned out that these trapped the staph bacteria in the vaginal canal, allowed them to proliferate in and around the tampon and elaborate toxins, and this brand new disease that had never been seen before, toxic shock syndrome, suddenly was described. And that's a staphylococcal disease associated initially with the use of tampons, but now we know there are other causes as well. One of the main toxins produced by staphylococci that causes toxic shock is called TSST1, toxic shock syndrome toxin number 1. And this is an enterotoxin. It actually acts as a superantigen. Superantigens bind the T cell receptor and the major histocompatibility protein on the surface of cells and cause massive release of cytokines from macrophages and T cells that are then responsible for the symptoms, say, of toxic shock syndrome. The whole idea of toxic shock elaborates from these cytotoxins that are produced. Staphylococci can also cause food poisoning. We've been talking about skin diseases of various sorts, but they can also be in your food, and they produce enterotoxins that are in the food. When you eat it, these make you sick. And the bacteria, if they contaminate the food, and you can imagine that someone may have Staph aureus on their hands. They're preparing food. They haven't washed their hands. They don't wear gloves. You now have Staph aureus in the food. If it's not kept cold, if it's at room temperature, which sometimes happens during food production or serving, the bacteria will grow. They will produce their toxins. And when you eat it, you will get sick. This typically happens with meats, including poultry, egg products, salads, milk, and dairy products. So if you ever have been to a buffet where there's lots of food spread out on the table, if it sits there for hours at room temperature, the bacteria can grow and make their toxins. Next time you go to such a function, make sure the people are wearing gloves. If they're not, you should just stick with the wine and don't touch the food. This disease, when you ingest these toxins, causes nausea, vomiting, stomach cramps, and diarrhea. And it's typically a rather rapid onset. So food poisoning, where you are eating food that has a toxin in it already, the onset is quick. So if you eat something and an hour or two later you're vomiting, you can say, wow, that could be a Staph aureus toxin-caused food poisoning. In contrast, if it takes a few days to get sick, that means the bacteria have to replicate in you before you get sick. So this is a rapid onset food poisoning caused by staphylococci, and then in 2 or 3 days you recover. And then you can blame the food handlers. All right, those are the staphylococci. Let's move to another coccus, streptococci, also gram-positive cocci, growing in pairs or chains, as you can see in this micrograph of the organism. We put them into 2 groups. There are a number of different ways to categorize streptococci. So if you decide by listening to this lecture you want to go read about streptococci, you may find other classifications, and you will say, Racaniello didn't know what he's talking about, but there are just many ways to do it. And so I'm going to tell you one way because we can't tell you all ways, and that is as follows. There are Group A streptococci, which cause a wide range of clinical diseases, and then there are Group B, which are leading causes of neonatal sepsis, that is disseminated infections, and meningitis. I like this because it's a very simple classification, and you can remember it. Just like the staphylococci, many people carry streptococci in them as well. This is ubiquitous in the human population. It's worldwide, it's in every country, and it's found on the skin and the nasopharynx, your nose and pharyngeal tissues, of about 20% of school-aged children. So if you go to any random classroom in the world, about 20% of those kids have this bacteria, the Streptococci, on and in them. And when you do carry Streptococci, usually you're okay. We say you're asymptomatic. You're a carrier and you can potentially infect other people, but you are okay. And this is a common occurrence. And when infection and disease occurs, it's usually in the form of, say, pharyngitis, and this happens in school children. So you may have a class full of kids, 20% of them have Streptococci. You put them together, they infect other kids who don't have the bacteria. They may get pharyngitis. We also call it strep throat. This is a rather painful throat infection. And if you get a throat infection and it's really hurting, it's a good possibility that it's strep throat caused by Streptococci. The bacteria may also infect the skin and soft tissues, causing lesions that are called pyodermas. You can see one here on the young lady's arm. Pyodermal infections usually require a break in the skin to introduce the bacteria, similar to the story with Staphylococci. All right? So in the pharynx, you don't require a break, but on the skin here, a break in the skin. If you're a carrier, you get an infection. If you're not a carrier and you're unlucky enough to have touched someone who has Streptococci on your skin, you'll get a pyoderma. It's spread from person to person, again, by respiratory droplets produced by coughing, sneezing, or talking, or by direct contact, very much like the Staphylococci. Most of these Streptococci remain localized, either in your throat, if you have pharyngitis, or on your skin, a pyoderma. But over the last 10 or 20 years, we have seen an increase in strains of Streptococci that apparently can invade deeper tissues. And this is serious and a worrisome occurrence because these are life-threatening infections. Now, so the bacteria are not just staying in the throat or on the skin, but they're invading, they're moving systemically, and as you can see, all the different organs in this young lady can be infected, and these are serious infections. Group A Streptococci produce proteins that promote spread of the bacteria. These are exotoxins that we've talked about before and include proteases that digest proteins in the host, which would normally restrict the spread of the bacteria. Hyaluronidases. Hyaluronic acid is a component of the extracellular matrix. And this is digested by hyaluronidase. It loosens up tissues and allows the bacteria to spread. And DNases. There's often extracellular DNA in tissues that makes it very thick, and it's difficult for bacteria to move through this. They make a DNase which chops up the DNA and allows free spread of the bacteria. These bacteria also produce an enzyme called streptokinase. It's shown here in three-dimensional structure of the protein. This enzyme converts plasminogen to plasmin. Plasmin, in turn, degrades fibrin. Fibrin is a component of blood clots, and it restricts bacterial movement. So this is another strategy that the bacteria have to move around the body. The bacteria also produce streptolysins, two different kinds, S and O. These lyse cells of various sorts, including immune cells that are trying to get rid of the bacteria. So you can see the value of that for the bacteria. And the bacteria also are very good at avoiding phagocytosis. Remember, macrophage-like cells are trying to take up these bacteria to destroy them. The bacteria have on their surface an M protein. It forms a dense layer on the surface of the bacteria. It binds complement and inhibits uptake by the macrophages. The bacteria also have a capsule on the outer surface. This is another anti-phagocytic structure. So it has at least two ways to avoid phagocytosis and destruction. Some of the more serious outcomes of streptococcal infections, in particular group A streptococcal infections, are non-suppurative sequelae. This means fever without pus. Now you remember, an abscess is full of pus, dead neutrophils, and other things, but there can also be fever and infection without pus, and that's what non-suppurative sequelae means. One of the more serious is acute rheumatic fever. This is a syndrome that can occur from 1 to 4 weeks after the initial infection. So a child gets strep throat, and if you have a child that gets strep throat, you should worry immediately and that this sequelae is going to occur, rheumatic fever, because it can be life-threatening, way beyond the sore throat, so get the infection treated. This infection involves carditis, infection of the heart, polyarthritis, infection of many joints, chorea, subcutaneous nodules, and erythema marginatum. The inflammation of the heart is the most serious issue here. This can cause scarring of the heart valves. So these bacteria infect the heart, the valves get scarred, and for the rest of the child's life, they have heart issues because of this. And this can also kill them. It can be a fatal infection. Why does this happen? It happens to be an autoimmune disease. We make antibodies against the bacteria as they're growing in us, but some of those antibodies also recognize our heart tissue and begin to attack heart tissue and destroy it. This doesn't happen in everyone, and it's probably a consequence of your particular immune makeup, but that's why it's an autoimmune disease. Rheumatic fever after strep infection with group A streptococci. Another important Streptococcus is Streptococcus pneumoniae, a specific species of Streptococci, also known as the Pneumococcus. This is a famous organism on so many levels. Here it is in this picture. It is usually cocci in pairs. And this bacterium was famous in research and medicine. In research, it's famous because it was used to show that DNA is the genetic material in the 1940s in a classic experiment. It's also famous because it's a very common causative agent of community-acquired bacterial pneumonia, pneumococcal pneumonia. What do we mean by community-acquired? You go out in the community and you get it from someone else as opposed to hospital-acquired, for example. So this is the most common cause of that kind of pneumonia. Over 1 million deaths every year throughout the world caused by Streptococcus pneumoniae. So this is a serious infection. There used to be many more, but we learned how to control it with antibiotics. Humans are the reservoir for this bacterium. Some of us harbor them and others do not. If you happen to not have a pneumococcus in you and you are infected from someone else, you may be able to deal with the infection readily, you may clear it and never get sick, or you may develop pneumonia, lung disease. And the outcome really depends on many factors, including the genetic makeup of your immune system, whether you have any sort of disease that would predispose you to lung disease, such as another type of lung disease, or if you're a smoker. This often is a predisposing factor. And finally, we think that the strain of pneumococcus also makes a big difference. Some are virulent and others are less so. This diplococcus or pneumococcus is spread from person to person by respiratory droplets, very much like other streptococci. So again, a carrier here maybe doesn't have disease, is speaking with someone who doesn't have the bacterium, who may be older and compromised in some way, and that person can acquire the bacterium and develop pneumonia. So initially you inhale the bacteria, it colonizes your nasopharynx, the upper part of your respiratory tract, and then it may replicate and spread down into the lungs, and there cause pneumonia. Conditions that decrease your cough reflex are important for susceptibility to pneumonia. Coughing is an important way of expelling organisms in your mouth and nasopharynx. We also have something called a ciliary elevator in our respiratory tract. Our respiratory tract is lined with cilia. These are tiny hairs that constantly move, and their function is to take any particles that you inhale and bring them back up again. So your tract is lined with these cilia, and it's also covered with mucus. And if you inhale a particle, one of your reactions may be to cough and expel it. Another thing that may happen if it's not very large is the ciliated elevator may bring it up, goes into the back of your throat, and then you swallow it, and it gets digested in your stomach. You do this routinely. Whenever you swallow, you're actually swallowing material that's come up from your respiratory tract. Some people spit instead. You know who they are. They're walking around the street spitting, which is kind of a gross habit. Better is just to swallow it and let it get digested. Anyway, if you smoke, you inhibit that ciliary elevator. You slow it down. And so that's why smokers in particular have more predisposition to respiratory diseases such as this one. The pneumococcus carries out a classic struggle with phagocytes. The phagocytes want to eat it. Of course, the bacteria don't want to be eaten. They want to multiply and survive to infect another day. So we have a battle between these two. The bacteria have a thick capsule that makes them resistant to phagocytosis. This is a nice illustration. Capsule makes the bacteria bigger, and it also has specific chemical interactions with the macrophage. The host, on the other hand, has a protein called C-reactive protein, CRP. This binds the teichoic acid in the outer membrane, the outer peptidoglycan layer of the bacteria. Remember, teichoic acids are stuck into that peptidoglycan. If you don't remember, go back and listen to me talking about it on one of the earlier bacterial lectures. This binding of the protein to teichoic acid activates complement, which is a host defense system that can then lyse the bacteria. We can treat strep pneumoniae infections. In fact, there was a time when they were uniformly lethal, but the development of penicillin shortly after World War I revolutionized the treatment of these infections and made people live who would otherwise die. But guess what? The overuse of penicillin and penicillin-like antibiotics has selected for resistant bacteria. And so now, if you get a pneumococcal infection, it's much harder to treat. Resistance involves penicillin-binding proteins that prevent the antibiotics from working. We now have vaccines that can be used to prevent infections, and there are two I want to tell you about. One is a pneumococcal polysaccharide vaccine for adults. So if you are an adult and you were never immunized, you should get this so you don't develop pneumonia caused by this bacterium at an older age when your lung functions start to decrease. This vaccine will protect you against 23 different serotypes. There's also a vaccine for kids. You should give it to them. It's called the pneumococcal conjugate vaccine. It contains 13 different serotypes of pneumococcal antigens linked to protein carriers. So give it to your kids and they won't have to get the vaccine when they get older because they will be protected. So today we have talked about two kinds of gram-positive cocci that cause a variety of skin and systemic infections, the Staphylococcus and the Streptococcus, diagrammed here. And after listening to all of this, I hope that you'll know the structure and virulence of factors produced by both Staphylococci and Streptococci. You'll be familiar with the pathogenesis and the epidemiology of diseases associated with Staphylococci and Streptococci. You should know which drugs can be used to treat staphylococcal and streptococcal infections and the problems associated with treatment, and you should become acquainted with the available streptococcal vaccines. Thanks for listening, and stay tuned to hear all about bacteria in your intestines. Hello, and welcome to Enteric Bacterial Diseases. We're going to explore a few different kinds of bacteria that infect the intestinal tract. And after you've listened to this lecture, you'll know the structure and virulence factors produced by bacteria that cause enteric infections. You'll be familiar with the pathogenesis and epidemiology of enteric bacterial diseases associated with Vibrio, E. coli, Shigella, and Salmonella. And you should know how to treat these enteric bacterial infections. First, we're going to look at a condition called secretory or watery diarrhea, which you and I have had for sure. Maybe not caused by bacteria, could be viral, but this is going to be different from diarrhea we talk about later, which is bloody diarrhea. All right, secretory watery diarrhea. And we're going to look at members of the Enterobacteriaceae and the Vibrionaceae. They're both gram-negative rods, two different kinds of bacteria, one shown on the left, the E. coli, and the one on the right, the Vibrios. These bacteria are transmitted to humans in contaminated food and water. Contaminated with what? Fecal material, of course, either human or from another animal. So in the end, it's all about keeping your food or water clean, which we seem to have problems doing. These bacteria that cause watery diarrhea adhere to the epithelial cells of the gastrointestinal tract. We showed in a previous lecture a slide showing the structure of the wall of the gastrointestinal tract. It's lined with epithelial cells, which you can see at the bottom of this slide. And the bacteria we're going to talk about sit on the surface of this epithelium. They don't go in, they don't invade, they're not invasive. They sit on the top and have their effects right there, and they adhere via their pili. And those are composed of surface adhesions. And again, if you don't remember what pili are, go back to the basic bacteria lecture. What these bacteria do is they make toxins. The toxins get into the epithelial cells lining the gut, and they cause problems. They reduce sodium absorption, and they increase chloride secretion. They disturb the homeostasis of sodium chloride balance, and that leads to water coming out of the cells, and there you go. There's your watery diarrhea caused by toxins. Very easy to treat. You don't need antibiotics. You just drink lots of fluids. Presumably water, or perhaps some water-based fluids with electrolytes would be fine, but just need oral rehydration therapy to replace what you're losing. Otherwise, you get dehydrated and you have more problems. Let's talk about Vibrio first. Vibrio cholerae, a cause of watery diarrhea. This bacterium is never a member of the human microbiome. It is not part of our normal flora. It is a normal inhabitant of the environment, in particular coastal estuarine waters. They live in close association with phytoplankton in the waters. And so they're living there, and they're very happy to be living there. So why do they infect us? Well, sometimes we enter this ecosystem, or the organisms contaminate our drinking water or food. So somehow they can get into our food or drinking water. Typically when sewage systems aren't working properly, Vibrio can get into our water supply, and then they infect us and cause problems. Cholera is the disease caused by Vibrio cholerae. It is the paradigm for secretory diarrhea. It is upon which everything is based, our knowledge understanding this condition. Vibrio cholerae encodes two toxins that are important for causing watery diarrhea. Once you ingest the bacteria, they get into your small intestine. They make cholera toxin, which is shown on the right of this slide there, CTX. It's composed of a subunit, which is the effector part, and the 5B subunits. We talked about this in a previous lecture. And a toxin co-regulated pilus. That means the other toxin, the toxin co-regulated pilus, is made at the same time as the cholera toxin. And this pilus allows the cholera to adhere to the surface of the mucosal epithelium, which is shown in the diagram on the right. So you have Vibrios, which are curved bacteria with a flagellum. They enter the intestinal tract, they make this pilus to adhere, and they produce cholera toxin at the same time. So what does cholera toxin do? It binds to a receptor on the surface via the B subunits. It's internalized, and the A subunit is released into the cytoplasm of the cell. This A subunit causes an increase in intracellular cyclic AMP. It raises the levels. This causes the imbalance in sodium absorption, so it inhibits sodium absorption, increases chloride secretion. As a consequence, the cells try and adjust. They get rid of water. Bingo, watery diarrhea. Okay, so cholera toxin is what does this by messing up the sodium chloride balance. There you get your watery diarrhea. Very simple, straightforward effect. Now, this watery diarrhea is a consequence of the toxin. What it does, of course, it washes the cholera bacteria out of your intestine, and that helps to spread the infection. If you happen to be in a place where the sewage treatment isn't very good, you will excrete cholera bacteria into the sewage, and it will then infect someone else via contaminated food or water. The challenge with cholera is to prevent dehydration. We say the watery diarrhea associated with cholera is a prodigious diarrhea. when you have cholera, you constantly excrete watery diarrhea. It's not like once an hour or once every 2 hours. It's continuously, prodigious. And if you didn't take in water or electrolytes, you would die of dehydration. And many people do, especially young kids who can't take in enough fluids. So preventing dehydration is how you treat this. You don't use antibiotics. You just give people lots and lots of fluids. And you have to give them a lot, because as soon as you give them a glass of water, it comes right out again. And you got to keep doing this over and over to prevent them from dying. In some countries, they've made a marvelous invention called a cholera bed. They're shown here. I probably don't have to describe them to you. It is a bed covered with a sheet of plastic. You lie on the bed, you're sick with cholera. There's a hole in the middle of the bed. You just lie there with continuous watery diarrhea. There's no point getting up and going to the toilet. In a facility like this, there are many, many people with diarrhea. They couldn't have enough toilets anyway, but you would just sit on the toilet all day because you have constant diarrhea. Instead, you lie in bed, and the watery diarrhea comes out of you, goes into this hole, and underneath is a bucket that catches the watery diarrhea. Modern medicine, cholera beds. It's actually just a way to collect the diarrhea, of course. What you really need to do is give people oral rehydration therapy. It's very effective. And you might think, well, isn't that the solution? Well, it's not always available. When there's so many people have cholera in a given region that may be economically depressed, it's not easy to find therapy to give people, in particular water or water with electrolytes in it. So you have to give this to people, and you have to make great efforts to get it and deliver it to people. Otherwise, people die. But if you supply oral rehydration therapy, you save their lives. Now, not every infection with Vibrio cholera is symptomatic. Not every infection causes watery diarrhea. You may ingest some Vibrios with contaminated water or food, and you may be fine. And you could be infected for a couple of weeks and be shedding the organism, potentially infecting someone else. That's called an asymptomatic infection. This is exactly what happened in 2010. You may remember there was a big earthquake in Haiti. The earthquake wrecked the sewage system, and so it wrecked many things, of course. And the United Nations decided to bring in workers from other countries to help out, which was a good thing. They brought in some workers from Nepal. It turns out these workers, or a good fraction of them, had asymptomatic cholera. They were shedding cholera in their feces. So they went to Haiti, and they defecated into a toilet, but the sewage system was broken, so their cholera contaminated the water supply. People in Haiti drank this contaminated water. It was a huge outbreak of cholera, as if they needed that in the midst of the earthquake. Now they had cholera. We didn't find this out until a bit after the incident occurred, that it was caused by these individuals from Nepal. Of course, they didn't do it on purpose, but many people were very upset about this, and they were mad at the United Nations, but it wasn't their fault. Okay, so that's Vibrio. No antibiotics, give people water, and they shall survive. Let's now look at E. coli in terms of its causing enteric disease. E. coli, Escherichia coli, very famous bacteria, is a member of the human gut microbiota. Most of the strains are avirulent. All the strains that we have in us, as long as they stay in the gut, they're okay. Now, occasionally, E. coli escapes the gut and causes other infections. You know, the way we are built is not ideal. We have our anus right next to our genitals. So material comes out in feces, it's easy to contaminate the genitals. And this is what happens in many people. They get urinary tract infections where E. coli escapes from the intestine and infects the urinary tract, can go all the way up to the kidneys and even spread beyond that. So these E. coli are just our normal flora, which aren't in the right place, in the urinary tract and elsewhere, and they can cause serious infections. We're not going to talk about these today, but I want you to know that the E. coli within you can sometimes cause problems within you. And those can spread to other people as well. What I'm talking about today are E. coli that come from other sources, either humans or animals, that are special strains that now have the ability to cause enteric infections. All right. Other E. coli strains, that's a picture of E. coli, by the way, they're rods, beautiful rods. Other E. coli strains, not from you, from other people or other animals, can be pathogenic. So these are spread by fecal-oral contamination. This can happen during food preparation. If a food preparer has this E. coli in them, perhaps they have an asymptomatic enteric infection with the strain that they acquired from somewhere else, they can contaminate the food and you will get it. Manure contamination is a big one. Cows have lots of E. coli strains in them, which don't hurt the cows at all, but if they get into people, they cause diarrhea. And often cow manure contaminates food of various sorts. So for example, crops are often irrigated with gray water that isn't so clean. It has cow manure in it and it contaminates the crops and they're not washed properly. You eat them and you get infection. So washing crops with contaminated water or just consumption of contaminated water that somehow contains manure from cows or even human feces, again from people who are infected with these unusual strains of E. coli. So the first strain we'll talk about, and there are several, is called Enterotoxigenic E. coli, or ETEC. There are lots of abbreviations we're going to be hitting here. We'll show you a table at the end so you can remember them all. ETEC causes traveler's diarrhea. Why does it cause that? You go to another country, within a day or two, you get diarrhea, watery diarrhea, after you've eaten some local food. That's traveler's diarrhea. It's annoying, but it happens. It affects children and adults all over the world. You've probably heard of it. It's caused by ETEC strains of E. coli. These use fimbrial or pili adhesions to bind to enterocytes in the small intestine, very much like the Vibrio that we've talked about. And these strains, the ETEC strains, make two enterotoxin. An enterotoxin is simply a toxin produced by the bacteria that affects your enteric tract. The ETEC strains of E. coli produce an LT enterotoxin. This is very much like a cholera toxin. It gets into the cell, causes increased amounts of cyclic AMP. And what does it do to sodium levels? Pumps it into the cell, pumps out chloride. There you go. You get your watery diarrhea as the cell then pumps out water to try and compensate. LT enterotoxin. The other toxin is called ST. This causes cyclic GMP accumulation in cells, which also results in secretion of fluid and electrolytes, contributing to the watery diarrhea. So two enterotoxins, both of their effects is to cause an imbalance in the ion makeup of the cell, and the cell excretes water, and you get watery diarrhea. ETEC, E. coli, two enterotoxins. These bacteria are not invasive. They sit on the surface of your epithelial cells in your gut. They attach to the gut surface by the pili, and they sit there, make toxins, and you have diarrhea. They don't go in, they're not invasive. It's an important distinction. These are simply bacteria, very much like the cholera, Vibrio cholera, sits on the surface, they do not invade. Let's look at another type of E. coli. These are enteropathogenic E. coli, or EPEC. Hard to remember, ETEC, EPEC now, enteropathogenic. These also cause watery diarrhea, but these bacteria are slightly different. They do not have fimbriae to attach. They have other things. They have no ST, and they have no LT toxins. Remember, those were two toxins produced by the ETEC strains, which were important for causing the watery diarrhea, but the EPEC strains of E. coli do not produce those toxins. So how do they produce diarrhea? These EPEC strains of E. coli adhere to the intestinal cells via two proteins. One is shown as a helical protein in the first panel of the illustration, and the second is via an intimin adhesion, which is this small blue box. And these cause the bacteria to adhere very tightly to the epithelial surface, and we think that binding changes the surface, and that causes the diarrhea. We call this adhesion an effacement, and it's a deformation of the villi caused by bacterial attachment, and that causes diarrhea, not the elaboration of toxins as with the ETEC strains. All right, so those are enteropathogenic E. coli. The next strain that we'd like to talk about are called enteroaggregative E. coli, or EAG-EC. These are strains only found in people And I mention this because these are frequently seen in AIDS patients, so these kinds of diarrheal diseases are associated with immunosuppression. Enteroaggregative E. coli have fimbriae, as you can see in this picture, which aggregate cells in culture, and that's why they're called enteroaggregative strains. They bind to the intestinal mucosa. They cause watery diarrhea without fever. They are non-invasive, as are the other strains. They do produce a number of toxins. One of them is a hemolysin, and they produce an ST enterotoxin, similar to that of the ETEC strains. The ST enterotoxins cause increase in cellular cyclic GMP, and this results in ion imbalances leading to watery diarrhea. Okay, so those bacteria, the Vibrio and the various strains of E. coli, cause watery, non-invasive diarrhea. Let's now turn to enteric bacteria that are invasive. They go beyond the surface of the epithelial cells. They invade the submucosal and they become systemic. And they also cause bloody diarrhea, which is also known as dysentery. So when you have blood in your diarrhea, it's no longer watery diarrhea, it is dysentery. The intestinal pathogens that do this structurally damage the large intestine, and they invade the mucosa, and that damage and invasion leads to bloody diarrhea or dysentery. So bloody diarrhea or dysentery, 30 passages a day, not as much as cholera, but 30 a day is still a lot. So it is stools with blood, mucus, and pus in them. That's the definition of dysentery. These are life-threatening infections. They require treatment with antibiotics. It's not sufficient to provide water rehydration, because here the bacteria are in your bloodstream, and you can have all the water in the world. You're not going to get rid of those bacteria unless you take antibiotics. So they need serious treatment. And we're going to talk about two different genera of bacteria that cause bloody diarrhea and dysentery. First, the Shigella, shown on the left here. And again, we're back to E. coli, but these are, again, different strains of E. coli that cause invasive diarrhea. So you see, E. coli can be very different depending on what genes it has. The first is the entero-hemorrhagic, or EHEC strains of E. coli, and the second, the entero-invasive strains, or EHEC strains of E. coli. So the Shigella come in 4 species: Shigella dysenteriae, Shigella flexneri, Shigella boydii, and Shigella sonnae, and these are all capable of causing dysentery. These bacteria are spread from person to person via contact or by contaminated food or water. And you don't need a lot of these bacteria to cause an infection. Only 100 or 1,000 are enough. So very minimal contamination of food, for example, or transmission of minimal numbers is enough to initiate an infection. So you ingest these organisms, they go to your intestine, and they invade through M cells. Now, M cells, you may remember from an earlier lecture about bacteria, are the cells that are interspersed throughout the other epithelial cells. And these cells are constantly sampling the lumen of the intestine to make sure there aren't any dangerous antigens there. So they take up things very actively and pass them through to immune cells on the lower side of the intestinal wall. Many pathogens take advantage of M cells to enter the host, and Shigella is one of them. They bind to M cells and they get passage through the M cell, and then they're released onto the underlying space, which is called the lamina propria. In this process, M cells are often full of macrophages that are there sampling antigens. They take up the Shigella in this case, but the Shigella are not destroyed by the macrophage. They escape the phagosome in the macrophage, which has taken them up, get outside, and they can spread elsewhere. But this transitory experience with the macrophage causes the release of cytokines from the macrophage. That causes inflammation, of course. The cytokines recruit other cells to the infected area, and all of this causes the symptom of infection. These Shigella, by the way, which are shown as the red oblong bacteria here can then invade epithelial cells from the bottom. So they've come in through the M cell, they're in the lamina propria, and now they go back into the epithelial cell from the bottom. They ride the host cytoskeleton up to the surface again, and now they're back on the surface of the epithelial cells. And they can go through multiple cycles of reinfection and being brought to the top again. Eventually, this destroys M cells as well as epithelial cells. The cells come off. So you end up having erosion of the gut wall or an ulcer. So this is a physical removal of cells as a consequence of bacterial inflammation. You have an ulcer, and in the process of doing this, many other immune cells have been trying to clear the infection, including neutrophils, major defense cells for many bacterial infections, as we've mentioned before. These cells die in the process and they accumulate in the stool, and that's what provides the pus for the stool, the presence of all these dead immune cells. So the lesion in the wall, the dying neutrophils, that gives you the blood and the pus in the stool, part of the dysentery. One of the species of Shigella produces another toxin called Shiga toxin. And this toxin has a specific effect in our cells. It cleaves the large ribosomal subunit, the 60S subunit, and that stops protein synthesis. So that basically kills the cells and is partly responsible for the cells leaving their position in the epithelium and causing that ulcer. You do need to take oral fluids to take care of a Shigella-induced dysentery. You don't need intravenous fluids. You can just take these by mouth. You don't need to be treated with antibiotics, as these are not invasive organisms. You can use antibiotics, but there's a lot of resistance out there, and they're not effective. Let's move to E. coli, one kind of E. coli, Hemorrhagic E. coli, or EHEC. Now, these are strains of E. coli that are frequently in the news, so you may have heard of them. EHEC E. coli. They cause non-febrile, no fever, bloody diarrhea, and hemorrhagic colitis is the name of this syndrome when caused by EHEC strains. So how does this work? These are strains of E. coli that produce toxin. They produce a Shiga-like toxin, which we've heard about before, can cause ion imbalances leading to water secretion. And there are several names for these strains as a consequence. Besides EHEC, they're also called STEC, which means Shiga-like toxin-producing E. coli, or HUSEC, hemolytic uremic syndrome-associated enterohemorrhagic E. coli. As we'll see, that's one of the syndromes associated with infection, or virocytotoxin-producing E. coli, VTEC strains. All these are the same name for EHEC-type strains. They're often classified according to the O antigen. Here we're showing lipopolysaccharide, which is a component of the outer membrane of these gram-negatives. You may remember this. If not, you can go back to one of the earlier lectures in which we talked about this in detail. One of the components of LPS is the O antigen, which you can see at the top there. And the O antigen is how we categorize many of these strains. And so, for example, you probably have heard of E. coli 0157H7. This is a strain that's caused many outbreaks of dysentery, and it's often acquired from cows. If you eat undercooked hamburger, you can be infected with an 0157 strain. The cows have this bacterium in their intestines. It does not harm them whatsoever. But if it gets to you somehow, either via the meat can easily be contaminated with cow feces during the preparation. If you don't cook it enough, you don't kill the organism, you will get infected. You will get the diarrheal disease from it. You can also get it from contaminated water. Again, if the water is contaminated with manure in some way by improper preparation, you will get this disease. 0157 is a specific kind of O antigen. So now you know what this means. The lipopolysaccharide, those O antigens at the very top in the previous slide, that's what is categorizing this as 0157. And if you're interested, the H7 is a flagellar antigen. The flagellum is the molecule that propels these bacteria around to move. This is one of the proteins of the flagellum, the H protein. So now you know that these EHEC strains are categorized by lipopolysaccharide and flagellar antigens. These bacteria may cause something called hemolytic uremic syndrome, and this is accompanied by hemolytic anemia. The red blood cells are lysing, so you become anemic, you have not enough red blood cells, thrombocytopenia, and kidney failure. These are all associated with these EHEC infections. A very large outbreak of an EHEC strain occurred a number of years ago in Germany, and these were caused by a slightly different strain, a brand new one that hadn't been seen before, O104H4. So now you know exactly what the O and the H antigens are. This caused a big outbreak in Germany of foodborne disease, and it was traced to contaminated vegetables. It was actually an E aggregate EC strain. Now, we talked about the E ag EC strains as causing diarrhea in AIDS patients. Somehow, this E ag EC strain acquired a gene for the Shiga toxin, which causes the watery diarrhea, producing this new strain O104H4. It was present on vegetables, probably via contamination with dirty water, and it wasn't washed properly. And if you eat uncooked vegetables, and you know people do like to eat their carrots uncooked, and even cucumbers and sprouts, particularly sprouts, are never cooked. And so these are common ways to get this infection. But this is an interesting case where the Shiga toxin had been newly acquired by one of these other strains. In the bowel, these EHEC strains produce proteins that allow them to attach and efface like the EPEC strains. So not all of them produce toxins. This particular strain in the German outbreak had acquired a Shiga-like toxin, but most of the EHEC strains simply attach and deface the surface of the villi, and that causes the diarrhea. The production of a Shiga-like toxin, if it's present, may be responsible for a cytokine response in the mucosa. You can get profuse bleeding, and this is caused by interaction of the cytokines that we produce with the Shiga toxins, which damages the blood vessel. And a major complication, as I mentioned, is this hemolytic uremic syndrome. This is a combination of damage to small blood vessels. If this happens in the kidney, you have kidney problems. And if you have extensive lysis of red blood cells, the remaining pieces of the red blood cells get hung up in the kidney, further complicating kidney function. These strains can also invade the brain, where it can cause thrombocytopenic purpura. And the Shiga toxins are on phages. And again, the Shiga toxin originally seen in Shigella, causing the loss of water from the intestine. These are on phages, so they're mobile. These toxins can spread from bacteria to bacteria, and that's presumably why the E104 German strain arose by infection with a phage that delivered the Shiga toxin gene at some point. These toxins are taken into cells and inhibit protein synthesis. Now let's move to Salmonella. These are another strain of enteric bacteria that cause focal infection of the vascular endothelium, may cause bloody diarrhea, as do some of the E. coli strains. These bacteria infect not just the gut, but a variety of other organs as well. They can infect the bones, for example, in patients with sickle cell disease causing osteomyelitis. Infections with Salmonella are associated with nausea, vomiting, and diarrhea. And then another consequence with different strains is called typhoid fever, in which we have systematic spread of the bacteria. These Salmonella strains are common members of the microbiome of many animals. For example, chickens, cows, and reptiles. Pet turtles often have these strains of Salmonella in them. The turtles are fine. In fact, all of these animals are not affected by their presence, but there have been outbreaks of Salmonella disease in children caused by contamination from pet turtles. You buy a pet turtle at a pet shop, you bring it home, and you have a Salmonella infection. Eggs are often contaminated with Salmonella, and there have been foodborne outbreaks of infection from egg products. Typhoid fever, another manifestation of Salmonella infection, is often transmitted from a human carrier. This can involve transmission via food or water. A very famous example of this kind of infection, typhoid fever, is Typhoid Mary. She was a carrier of the bacterium. She was not sick, and she was involved in food preparation and infected many, many people until it was discovered that she was the source of the infection. So Typhoid Mary lived many years ago, and the solution was to put her away in a room and not let her interact with anyone, which is not really satisfactory, of course. But now we understand what's happening, and we can take other measures to minimize transmission from these carrier individuals who still exist. So as you might guess, these infections are acquired by fecal-oral contamination. In the case of Typhoid Mary, for example, the bacteria, she's excreting them. If she doesn't have good hand hygiene, she's going to contaminate food preparation. The bacteria make their way to the intestine. They penetrate the mucosal barrier. They enter M cells as well as epithelial cells, and they're carried through to the underlying tissues by endocytic processes. Bacteria make their way to the lamina propria, which is, again, that space underlying the epithelial sheet. In the process, sodium chloride imbalance is introduced, so that diarrhea results. And the bacteria then spread from the underlying spaces to the bloodstream. So this is not something that we see with the watery diarrhea strains, the spread from the initial site of infection. They're confined. But these Salmonella strains can spread typically from the lamina propria. The bacteria enter mesenteric lymph nodes, and then from there enter the bloodstream, where they can spread. So these infections are associated with gastroenteritis and the production of cytokines and an influx of neutrophils, which attempt to clear the infection. And of course, as the neutrophils die, they're released into the stool, which has pus in it. Typhoid fever is a kind of infection that involves dissemination of bacteria from the intestine using the mechanism that I just described. But there are also non-typhoidal systemic infections with Salmonella that are caused by food-acquired bacteria, which go beyond the lamina propria in the intestine, get into the bloodstream, and spread to other tissues. These are not considered infections of typhoid strains of Salmonella. They are non-typhoidal. So let's look at how typhoid-causing strains survive and infect. So we take them up into the gut, as you can see on the top there. There's an incubation period shown by the dotted line where the bacteria invade the small intestine, move to mesenteric lymph nodes, and then enter the lymph system. They can replicate in macrophages in various organs. So once they're in the blood, they can spread to various organs. They're taken up into the organs. They can replicate in macrophages of the liver, the spleen, other lymph nodes. This is an asymptomatic phase, part of the incubation period. Then these bacteria are released from the phagocytes into the bloodstream. This initiates a septicemia or a bacteremia, and that can, again, spread the infection to other organs. And that release is associated with high fever, the typical sign of typhoid fever. Once the bacteria are in the blood for this second phase, they can invade the gallbladder. And from there, they can go on and reinvade other tissues. But the gallbladder is an important site because this is often a place where chronic infection is maintained. From the gallbladder, of course, the bacteria can get out through the bile duct into the intestinal lumen and be shed, or they can reinvade tissues, cause ulceration of Peyer's patches in the gut mucosa, be released into the stool, and can be present in the urine as well. So there's a cycle here that you can see from the initial infection, multiple releases into the bloodstream and other organs, and then release and reinfection as well. This gastroenteritis can be treated with antibiotics, but the use of fluoroquinolones in treatment of this disease is controversial. And if you read the literature, you will see people who believe you should treat or you should not. The systemic non-typhoidal infections, non-typhoidal meaning not Salmonella typhi, these require antimicrobial therapy. Of course, in all cases when there's diarrhea, you also have to have oral rehydration therapy. Typhoid fever itself, caused by Salmonella typhi, is very difficult to treat. People who are carriers of the infection the bacteria cannot be eliminated with antibiotic treatment. So they have to be very careful not to contaminate others with the bacteria in them. So we've now gone through a series of bacteria of various types that cause intestinal infections. Let's summarize this so that you can see it all together and try and make sense of it. And these tables will be a good place to study this if you need to remember them. So we've talked about watery diarrhea or secretory diarrhea. This is, again, when you have a lot of watery diarrhea, no blood, no pus, no tissue invasion. The site of infection is typically the small intestine, and examples are Vibrio cholerae and the ETEC, EPEC, and EAG, EC strains of E. coli. All of those can cause this watery diarrhea. Dysentery, there's much less volume, and there's also blood, mucus, pus in the diarrhea, and these bacteria can invade and move beyond the initial site. This is typically in the large intestine, and this can be caused by Shigella. There are also parasites that can cause this infection, and if you're interested, we can talk about that in the parasite lecture. We have hemorrhagic colitis caused by EHEC strains of E. coli. Again, copious liquid-like diarrhea, but this has blood in it, although there are no cells, no tissue invasion. This happens in the large intestine. And again, bloody, watery diarrhea, copious diarrhea with blood or pus in it, and tissue invasion. This happens in the ileum and the colon. And we talked about strains of Salmonella that can do this. There are also other bacteria, including Campylobacter and Yersinia, that can accomplish similar diseases. So this is a summary of everything on one slide. You can see the different ETEC, EPEC, EAG, EC strains, the EHEC strains of E. coli, Vibrio, Shigella, and Salmonella, and exactly what kinds of disease they're causing. Again, we have looked at gram-negative rods in this lecture in terms of their effect on the enteric tract. We've looked at Vibrio cholera, we've looked at Escherichia coli strains, and Salmonella. And I hope after you've listened to this lecture, you know the structure and virulence factors produced by bacteria that cause enteric infections. You should be familiar with the pathogenesis and epidemiology of enteric bacterial diseases associated with Vibrio, with E. coli, Shigella, and Salmonella. And you should know how to treat these enteric bacterial infections. Thanks for listening, and I hope you enjoyed learning all about various bacteria. And I hope after you've listened to this lecture, you know the structure and virulence factors produced by bacteria that cause enteric infections. You should be familiar with the pathogenesis and epidemiology of enteric bacterial diseases associated with Vibrio, with E. coli, Shigella, and Salmonella. And you should know how to treat these enteric bacterial infections. Thanks for listening, and I hope you enjoyed learning all about various bacteria.